All right. Uh, Continuing with chapter 6 here, looking at cells. Next, we're going to look at mitochondria and chloroplasts. So, concept 6.5 mitochondria and chloroplasts change energy from one form to another. So, mitochondria is the site of something that we call cellular respiration. And this is a metabolic process where we take organic molecules that could be lipids, it could be protein, it could be carbohydrate, and we use that to strip off electrons and pass them on to oxygen in the system that produces ATP. Now, chloroplasts are serve a very similar function in plants and algae, in that they are the sites of photosynthesis. And photosynthesis is very similar to cellular respiration, except for instead of taking electrons off of organic food and dumping them onto oxygen producing ATP, what we do is actually take electrons off of water, energize them through sunlight, then combine them with oxygen, and in the process produce ATP. So they have very similar systems, and they're structured somewhat similarly because of that. And the last thing in this section that we talk about is peroxisomes, which are special organelles that are meant to carry out something, an oxidative process that is very similar to what's happening up here, except for we don't produce ATP. All right, so both chloroplasts and mitochondria have a lot of similarities to bacteria. So both this organelle and this organelle are double membrane, and most bacteria are double membrane. Both mitochondria and chloroplasts have their own small pieces of DNA, and notice I said DNA, not RNA, and it's circular in nature. So a small circular piece of DNA in mitochondria, small circular piece in chloroplasts. Just so happens that DNA in bacteria tends to be circular. The ribosomes in bacteria are very different than our own ribosomes. And oddly enough, you look inside mitochondria and chloroplasts, you will find other forms of ribosomes that are very similar to bacterial ribosomes. Inside of our cells, mitochondria and chloroplasts have the ability to reproduce themselves. So you make more mitochondria, not de novo, but you grow one mitochondria bigger and then you split it into two. Same thing is true for chloroplasts. That is essentially how bacteria grow. All these facts lead to the theory called the embosymbiont theory. This theory suggests that an early ancestor of eukaryotic cells engulfed an oxygen-using non-photosynthetic prokaryotic cell. This could have been a parasitic bacteria at some point, but over a long period of time produced a more cordial relationship. These embosymbionts that live within these other cells would have evolved into current day mitochondria. The same idea is true for photosynthetic bacteria. So there are photosynthetic prokaryotes or photosynthetic bacteria that look an awful lot like chloroplasts. So here's an idea. We have some larger eukaryotic precursor cell And at some point, it gets this bacteria living inside of cytoplasm, and over time, that turns into mitochondria, or you have then this early eukaryotic cell bringing chloroplast, or bringing a photosynthetic bacteria inside, and ultimately it would evolve into a chloroplast. Now, other pieces of evidence that help with this theory: there are, in fact, certain types of bacteria that can invade our cells and grow within our cells. One is a tick-borne disease called rickettsia, and if you get it, it's not good news. It produces a disease called Rocky Mountain spotted fever, and these bacteria live in the cytoplasm, reproduce, ultimately killing us. However, if you look at the structure and genome and a lot of the parts, they are very similar to what's left over in mitochondria. So you can imagine that ultimately, one of these cells got in, didn't kill the host cell, and over time, as it was living inside this host cell, it needed less and less and less of its own material. So it would lose parts of itself that it didn't need. And it would also start to specialize in the activity of producing energy. So mitochondria produce energy, produce ATP for the cell. And you could say that it does this in exchange for a nice, warm, cozy place to live. Same thing can be true of um, photosynthetic bacteria. There are, in fact, um, animals out there that currently host actual photosynthetic bacteria or algae within their cells or within their bodies in order to siphon energy off of them. They have their own internal farm. And you could say that chloroplasts are just a more evolved example of this. Specifically for chloroplasts, they look a whole lot like this bacteria called blue-green algae, or blue-green bacteria. It is a prokaryotic organism. All right, mitochondria are pretty much in all eukaryotic cells. Whenever you have an absolute, there's always a exception, but for the most part, they're always there. They have a smooth outer membrane, and then the inner membrane is very invaginated and folded into this all these folds that produce more surface area. This surface area is the site where we produce ATP. So the basic process of ATP, producing it within mitochondria, is we strip electrons off of something else, some type of organic material. We pass them through a chain in the membrane, which pumps protons from one side of the membrane to the other. And then we run those protons back through what's essentially a little water wheel pump that literally squeezes phosphate and ADP together to produce ATP. Again, don't worry about too much about that right now. We'll go into that in more detail in future chapters, but as a sneak peek of what's happening here. So having lots of surface area is important, and all these invaginations produce that surface area. All right, so this produces a couple of spaces. We have an inner membrane space in between these two membranes, and then the inside, which is called the matrix. And those membranes are the site of ATP synthesis. Here's another zoomed in picture from before. Here we can see an electron micrograph. showing these invaginations. And here is a fluorescent micrograph of mitochondria. And you can see here is a fluorescent stain for DNA. So you can see mitochondria have their own DNA. And there's also another marker in here that allows us to see all these invaginations, all this area that makes up the matrix. Here is a three-dimensional model built from a series of micrographs similar to this one. So we'll take a look at it here. All these little channels produce one large internal region and then a separate second intermembrane region. So all these little canals are all connected to each other. Now, chloroplasts look very similar. You can see that they are double membraned as well, that the inner membranes produce these stack-like structures, which are very reminiscent of these mitochondria, but much more organized. And these internal stacked membranes, or thylakoids, are the site where photosynthesis takes place. Photosynthesis works a lot like energy production in mitochondria, in that it takes place on the surface of a membrane. That means the more membrane we have, the more energy producing molecules we can build. Part of this system is this green pigment called chlorophyll, which is involved in absorbing energy from light and transferring it onto electrons. Those energized electrons are then used to do work and ultimately produce ATP. So this internal region here, we have the thylakoids. They are stacked into these things called granum. The internal space is called the stroma. Again, a lot of words that I'm never going to test you on. I'm more interested in that you can visualize it or draw it or explain it by words. I'm not overly concerned with definitions like thylakoids, but just so you know that that is what they are called. All right, the final thing to talk about are these things called peroxisomes, and here's an electron micrograph of a peroxisome, membrane-bound structure. Here's a mitochondria and a couple of chloroplasts for size comparison. Inside of peroxisomes, we carry out reactions that could be dangerous to the rest of the cells. One of those is utilizing enzymes that transfer hydrogen from substrates onto oxygen. The hydrogens being moved is actually a side product. What we are doing is stripping electrons off of these compounds and moving them onto oxygen. This is the same reaction that occurs in mitochondria, but here we're not interested in producing energy. What we're interested in producing double bonds. So this is an area where we take longer hydrocarbons, such as fatty acids, we move them into here, and within peroxisomes we can add double bonds. And we do that by stripping away electrons and forcing neighboring carbons to share electrons. We strip those electrons off, transfer it onto water. This converts the water, or this produces something that we call hydrogen peroxide, which ultimately degrades back into water. Now, hydrogen peroxide is a toxic compound. We need to keep it contained. This reaction itself is not a safe reaction. It produces something called oxygen-free radicals, and we need to hide that.